Osmosis is a process where the water in a solution passes through a semi-permeable membrane, moving from a high water potential to a low water potential. This occurs when there is an uneven concentration of solute between the two sides, creating what we call a concentration gradient. Water potential in osmosis is determined by looking at the potential energy the water in a solution has compared to pure water. In other words, the potential or tendency the water has to move to the other side through a semi-permeable membrane, based on how the solute concentration relates to the water amount. The higher the solute concentration, the lower the water potential. To reach equilibrium, water moves towards the side with more solute. As seen on the screen, since the left side has more water compared to its amount of solute, it has a higher potential to move to the right side, which has little water compared to its amount of solute. Ideally, if equilibrium was reached and the solute concentration and water amount were proportional to each other on each side, it would look something like this. Now let's look at how osmosis occurs in animal cells. When an animal cell, such as a red blood cell, is exposed to a high solute concentration with a low water potential, the red blood cell is considered to have the higher water potential, so water is drawn out of the cell and into the solution. This shrinks and dries up the cell as a result. This type of solution is called a hypertonic solution. Equal water potential means that the amount of solute and water, both inside and surrounding the cell, is proportional. In the example on the screen, a medium amount of solute is present. However, as long as the concentration is the same on both sides of the semi-permeable membrane, it does not matter. It could have a high, low, or as I have here, a medium concentration of solute. Because equilibrium has been reached, there is no net movement of water, and the cell remains in its best condition. This is called an isotonic solution, and is the most ideal solution for animal cells. When a red blood cell is exposed to a solution with a low concentration of solute and a high water potential, this cell is the one with the lower water potential, and resultantly water is passed into the cell from the solution, puffing it up and oftentimes popping it. This is called a hypotonic solution. Osmosis in plant cells differs in some ways but is the same in others. Animal cells have such dramatic reactions to the solutions because they do not have a cell wall. Plant cells, on the other hand, do. Turgor pressure is the pressure exerted on the cell wall by the cytoplasm and helps to maintain shape. In hypertonic solutions, the loss of turgor dehydrates the cell but is not as detrimental as in animal cells. Additionally, this loss of turgor is what causes flowers and leaves to wilt. In an isotonic solution, plant cells simply remain the same, and the amount of turgor pressure is decent. However, in a hypotonic solution, plant cells thrive the most. The turgor pressure is strongest in this type of solution and gives the cell the most water possible to thrive. Too much cannot hurt it because the cell was built to withstand.